Yeah.
I didn't see you there.
Oh, hey guys! So you're probably wondering why I'm able to lift so much weight. Everyone knows that the reason anybody can lift weight is because we all possess tissue called muscles. The muscles I'm using right now are called skeletal muscles. They're different from smooth muscles which aid in peristalsis or from cardiac muscles that keep your heart beating.
So why exactly do skeletal muscles allow us to lift weights? Let us take you on a journey of skeletal muscle contraction.
But first, let's talk about the structure of skeletal muscles. Each muscle is attached to bones at both sides. For example, the biceps brachii, not to be confused with the biceps femoris, which is in the back of your thigh. The biceps brachii muscle is composed of two heads. The long head on the lateral side and the short head on the medial side. Both ends are attached to different bones. The origin is on the scapula, while the insertion points lie on the radius and the ulna. Both points are attached to the respective muscles and bones via tendons.
Okay, that's it.
I see you struggling there with those weights. You know, whenever I want to do a bicep curl, a signal from my brain is sent to my muscles. It sends it via the piano.
But let's pause on the mechanics and focus on the structure.
Each muscle is divided into muscle fascicles. These in turn are divided into the multinucleated muscle fibers. These run the entire length of the muscle. When you work out and trying to gain muscle mass, muscle fibers do not divide. You keep the same amount of muscle fibers unless there's a disease associated with it. Each muscle fiber is enclosed with a structure called sarcolemma. Instead, what increases the number of myofibril. That's what the muscle fiber is composed of. Now, let's focus on the sarcomere.
A sarcomere exists throughout the length of a myofibril. They are the reason a muscle has the ability to contract. One sarcomere exists between two Z discs. The working units of the sarcomere are called actin, that are thin filaments, and myosin, or thick filaments. Actin molecules are attached to Z disc directly. Actin molecules are attached to the Z disc, while myosin molecules units are attached to the titin proteins. The width of a myosin molecule is called an A band. The I band is the distance between the myosin molecules of neighboring sarcomere units. The end zone lies in the middle of the sarcomere unit, in between the actin molecules. This is the part that shortens during muscle contraction. Ready? Okay, so myosin molecules have a very simple structure. The light meromyosin connects to the heavy meromyosin, and the heavy meromyosin is simply called the myosin head. This is what
Now that we have explained the structure in much detail, we can finally focus on the mechanism of the skeletal muscle.
Once the electric signal or action potential reaches the end of the motor unit, the neuron secretes the neurotransmitter acetylcholine. This further propagates the action potential. The action potential travels along its sarcolemma. The sarcolemma has indentations called T-tubules, or transverse tubules. The action potential travels within these T-tubules towards the interior of the muscle, where the sarcoplasmic reticulum resides. The depolarization of the T-tubule membrane occurs. It causes voltage-gated calcium ion channels on the sarcoplasmic reticulum to open. It is sensed by a DHP receptor. This is communicated to the ryanodine receptor, which opens and causes a release of calcium ions.
Notice that the sarcoplasm is just like the cytoplasm, only that it is located in the muscle fiber, and the sarcoplasmic reticulum is like an endoplasmic reticulum with the same difference. It resides in the muscle and it serves as a calcium ion storage unit.
Is that it?
Along is called sliding filament theory. Calcium ion move toward the sarcomere. Troponin complex, which was mentioned earlier, has a higher affinity for calcium. Therefore, calcium binds to the troponin complex and changes its conformation, since troponin sits on top of the tropomyosin. With the conformational change with troponin, tropomyosin is also changed conformation. Also uncovers the binding sites, which are located on the F-actin molecule. Once the binding sites are free, the myosin heads from the myosin molecule intermediately attach themselves to these sites, creating cross bridges between the thick and the thin filament.
Whoopsie. Yeah.
The myosin head is capable of attaching itself to the actin by releasing ATP and phosphate. Notice that it is not ATP, it is adenine diphosphate, that is ATP. The bound ATP and phosphate attaches itself to the binding site, and this causes the myosin head to be a different non-relaxed conformation. Now that the head is attached to the binding site and the ATP and phosphate molecules are gone, now when the ATP and phosphate molecules are gone, the myosin head relaxes and pulls on a thin filament with a power stroke.
Yeah. Okay. This pulling of thin filaments on a macro scale is what we call muscle contraction. The H zone length is shortened and the opposing thin filaments on one sarcomere come closer. The thin filaments slide along the thick filaments as long as there is a supply of calcium ions that can be bound to the troponin. In order for the myosin heads to keep attaching to the thin filaments and pulling them, there also needs to be a steady supply of ATP. ATP binds to the myosin head, which acts like an ATPase, and it splits the ADP and phosphate. And this results in continuous sliding. And that is how, for example, I am able to pick up this weight.
After an intense workout, it would be pretty awkward if I could contract my muscles and not relax them. The reason calcium ions are still bound to the troponin is because the action potential is still propagated. Once the decision is made to relax the muscle, the action potential is ceased. Now that there is no depolarization of the sarcolemma and the T-tubules, there is no more calcium ions to release. The calcium that has been displaced earlier into the sarcoplasm is quickly picked up by calcium ion pumps, which reside in the sarcoplasmic reticulum. The question located inside of the sarcoplasmic reticulum facilitates storage of calcium ions until the next action potential is sent towards the neurotransmuscular junction.
Some people are able to run a marathon in 2 hours, but not be able to sprint a 100-meter dash in 10 seconds, or vice versa. Why are some people muscular with little to no effort, while others spend hours at the gym and just can't seem to bulk up?
The difference is associated with fast twitch fibers and slow twitch fibers. The fast twitch fibers are associated with muscular bodybuilders, while the slow fibers are the ones that predominate on marathon runners. This means that you are born with a predisposition to what kind of an athlete you might become in the future. If it's endurance-based, it's slow twitch. If it's strength-based, it is fast twitch.
The speed at which a muscle is able to contract relies heavily on the myosin to split ATP into ADP and phosphate. If ATPase capabilities are high and velocity is high, then we have a fast twitching muscle. The opposite is true in that if ATPase capabilities are low and velocity is low, then we have a slow twitching muscle.
Most muscles contain both types of muscle fibers, both in different proportion. However, one motor unit always has the same fiber type it works on. A typical person has a little more fast twitching muscles than slow twitching ones. Usually it is a 55 to 45% ratio.
Depending on the amount of fast twitching fibers, a person can gain muscle weight. It is due to strength training. The reason muscle grows is because of hypertrophy. As mentioned earlier, muscle fibers do not usually split. It is the myofibrils within them that split. More action and myosin are formed due to the need for extra strength.
As a person grows from infancy, it is normal to have a muscle fiber lightening. If muscle fiber splits, that's a problem. It's called hyperplasia and usually occurs during muscle cancer. However, under extreme endurance training conditions, it is also possible to grow new muscle fibers, although it is very rare.
So there are a lot of reasons why one can't seem to bulk up at the gym. Not just one's predisposition to the concentration of fast-twitching muscles. Educating oneself further in order to understand the mechanics of the human body will surely aid in the pursuit of a more Schwarzenegger-like body and a healthier lifestyle at that. Perhaps one day, when more research is done, people with muscle atrophy will be able to take myostatin pills and not suffer from muscle degeneration any longer. Also, on the other side, people will turn to these sources as a performance enhancer.
For now, we hope you enjoyed our video and don't take drugs.
Unless you're told to by your friendly neighborhood pharmacist.
Megastroid! I can't. You're probably wondering.
Depends on the amount of weight a person can lift.
And myosin molecules are attached to the tiny proteins.
Say the whole sentence again, so he can cut it out.
Again,
Depending on the amount of fast. Depending on the amount of fast. Sanford and some music.
We could.